Okay, this is my view of cancer research over the past 80 years. Before the discovery of oncogenes and tumor suppressors in the 70s, cancer researchers were really focused almost exclusively on the metabolism of the tumor cell, which differs quite a bit from normal tissues in which the tumors arise. Since the discovery of oncogenes and tumor suppressors in the 70s, people have largely ignored tumor cell metabolism until very recently. And we're really interested in understanding how oncogenic signaling networks impinge back on the control of tumor cell metabolism. This is the metabolism of the tumor cell that allows it to proliferate and grow in an otherwise hostile environment often. So our work in this area is focused on mTOR within mTOR complex 1. mTOR is unique in that it senses a variety of physiological and pathological signals pictured here, the variety of nutrients and secreted factors, growth factors, as well as the majority of oncogenes and tumor suppressors impinge on the control of mTOR complex 1. And we're really interested in understanding what mTOR complex 1 does under both physiological and pathological conditions to control anabolic cell growth and proliferation. Focusing in on cancer, we now know that mTOR complex 1, or mTORC1, is activated in approximately 80% of human cancers, and this is across nearly all lineages. And this is due to a large variety of oncogenes and tumor suppressors that lie upstream of mTORC1 and ultimately upstream of a small G protein switch that is responsible for the control of mTORC1, impinging on this small G protein called Rheb. So in recent years, we've been very focused on understanding what's downstream of mTORC1 within human cancers that might contribute to its role in cancer. Its best characterized functions are the inhibition of a process called autophagy, which is recycling of nutrients in the cell through the degradation of organelles and long-lived proteins. mTOR complex 1 also stimulates protein synthesis through the control of both mRNA translation and ribosome biogenesis. And these processes, while not fully understood, are really the two hallmarks of mTOR complex 1 function that are well established. In recent years, we've discovered that a large variety of metabolic processes are downstream of mTOR complex 1. These include the promotion of aerobic glycolysis, which is now recognized as a major source of biosynthetic precursors for growing cells. And this is really something that's also referred to as the Warburg effect in tumor metabolism, named after Otto Warburg, who originally discovered this in the 30s. mTOR complex 1 also drives de novo lipid synthesis, the oxidative phase of the pentose phosphate pathway, and de novo nucleotide synthesis. And through these processes, controls macromolecular synthesis, the synthesis of proteins, membranes, and nucleic acids, which is really all the building blocks required to make new cells. So that we think this is its major role in cancer, in that it links the oncogenes and tumor suppressors to the control of these metabolic processes. So briefly, just to illustrate how mTOR complex 1 does these things, I've kind of summarized all of these things on one slide here. We found a few years ago that mTOR complex 1 regulates glycolysis in blue, the pentose phosphate pathway in purple, and lipid and sterol synthesis, fatty acid and sterol synthesis in orange, through the control of the metabolic enzymes that drive those processes, so through the transcriptional control of those processes. So the genes we found to be induced by mTOR complex 1 are in red within the metabolic pathways in which their enzyme products control. So we found that mTOR complex 1 regulates these metabolic processes largely through two transcription factors: glycolysis here through HIF1 alpha, lipid and fatty acid and sterol synthesis through the sterol regulatory element binding protein transcription factors. And we also found that SREBP1 regulates the rate-limiting enzyme of the oxidative phase of the pentose phosphate pathway. And in doing metabolic flux studies, we've also found that mTOR complex 1 promotes metabolic flux through the oxidative pentose phosphate pathway. We think this is an important source of reducing power to drive anabolic growth, in that it produces NADPH, which is required for processes, biosynthetic processes such as lipid synthesis, but also for nucleotide synthesis. And another critical element of nucleotide synthesis is the production of the sugar ribose, which is added on to purine and pyrimidine nucleotides. And we now know that TOR also regulates purine and pyrimidine synthesis, at least in part through the control of the pentose phosphate pathway and ribose production. In a very recent study, we've also found that in addition to the control of metabolism through transcription factors, mTOR complex 1, through its downstream target S6 kinase, directly phosphorylates the rate-limiting enzyme of pyrimidine synthesis and really acutely stimulates the synthesis of pyrimidines through the phosphorylation of this enzyme called CAD. So just to summarize, we now know that mTORC1 really is a major link between growth signals, and these are growth signals from both physiological and pathological sources, such as oncogenes and tumor suppressors, and it links these growth signals to the control of the metabolic processes that underlie growth. So mTOR complex 1 is not required for these metabolic processes, but rather is required as a conduit between growth signals and these growth processes.